Well, hello. Welcome everyone to this webinar, Healthy Soils at Rio Fernando Park. My name is Ben Wright, and I work with the Taos Land Trust as a Land Projects and Education Coordinator. And presenting with me will be Amy Larsen, and she's a researcher at NMSU Sustainable Agricultural Science Center in Alcalde. I'm going to be presenting the broad picture of what we're doing for healthy soils here at Rio Fernando Park. And Amy will be giving some more detail on just the scientific basis and some of the theory behind some of the projects we're enacting here. I don't know if you all had a chance to see it, but fairly recently you were emailed some supplementary materials. They're not really necessary for the presentation, but more for just review later. And there's some just links and more information about some of the things we're going to be talking about. So don't forget to take a look at those. It might help you in the future. We'll get started. Thank you all for coming. So I'm sitting here, the backdrop behind me is actually part of Rio Fernando Park. Some of you may be familiar with this space, but basically this is a 20-acre piece of land in the town of Taos. This land is owned and managed by the Taos Land Trust. And the purpose of this land is to make available both a natural space and an agricultural space for members of the community. And on this map, you'll see this blue line here. That's the Rio Fernando. And over here is Baca Park. So that will probably orient many of you. The Rio Fernando, of course, starts up high in Taos Canyon and goes across town, goes through our property, goes through Baca Park, and then some neighborhoods, and ultimately joins the Rio Grande. So this river creates a natural wetlands area in this area. And so we are basically doing restoration projects on that area of land for natural habitat. So we're trying to support the bird species that live there, the beaver that live there, and many other assorted species that use wetlands habitat for their home. The pink track, those are trails around the property, and this property is open to the public, so feel free to come around and walk around and check things out. What we're going to be talking about today is more these areas, the pollinator habitat, grain field number 2, grain field number 1, and then this area, which will be a future orchard. But we're doing our healthy soils projects on all of these areas of land. We're building compost right around this area, and then we have vegetable growing beds here, which we're also using some of the compost, but it's less of a focus for the Healthy Soils project. So the purpose of this talk is to look at the projects that are happening on this land, but also give you the general information about some of these techniques so that you may be able to apply some of these applications on your own land. Healthy Soils is part of an initiative. It's a project that was sort of started by the New Mexico Healthy Soils Working Group. And they picked up on some information from the NRCS and just general concepts. A lot of projects were taking place in Australia and Africa and different parts of the world, like looking at how to improve soil health. So some folks got together and said, well, what can we do for New Mexico? So they pushed and they asked for money, and the result of that is that the Healthy Soil Act was enacted in April 2019, so a little over a year ago, and that led to the creation of the NMDA Healthy Soils Program. And of course, Taos Land Trust has been a beneficiary of that program through the Vigili Romo Acequia, and we are taking that opportunity to do the best we can to do soil restoration on Rio Fernando Park. So healthy soils, what does that mean? This is the short description, and it's based on these 5 principles. Some people say there's 4 principles because they kind of intermix a little bit, and some people up it to 6. But this is basically the grounding of it. Keeping soil covered, no bare ground. So just making sure that you either have something growing, that's the preferred method, or at the very least, some sort of layer of mulch. We're really keen on minimizing soil disturbance. So that's no-till techniques. We're also not interested in bringing in external inputs. So chemical fertilizers are out, chemical insecticides are out. We're working with compost that is built on site. We're attempting to maximize biodiversity both above and below ground. So that's plants growing above, plants and animals and insects above, and microbial diversity below ground. And we'll get quite a bit deeper into that aspect in particular. Maintaining living roots, so living cover crops, keeping something growing on the land. I mean, they say nature abhors a vacuum. So we're trying to fill it with growing things, because that really, life feeds life, and that the growing mat feeds the microorganisms underneath. And then we're also integrating animals, and we're looking at the full gamut of animals: grazers, animals, insects, pollinators, soil animals, and the whole microbial community underneath. In particular, we are applying these principles on the land at Rio Fernando Park. And we'll go into more detail about all of this, but looking at the soil processes and really our goals for this project are to reduce the inputs, as I said, and really create a healthier ecosystem that we can both do restoration projects and also grow crops that we desire, plants that we desire, natural habitats that we desire. So really we're attempting to change the degradation that has occurred on this land over the last 30 or so years. This is a former farmland that has basically been heavily used and then abandoned. And we're trying to re-steward the land in ways that will sustain long-term goals such as regenerative agriculture and ecological restoration. And we'll turn it over to Amy to go into some more details about what some of these processes may be.
Thank you, Ben. So I think, as most people kind of know intuitively, the soils are just a hugely complex kind of ecosystem. There are lots of processes that go on that interrelate, and they influence each other. They can basically be broken down into 3 categories. You have your biological processes, and those include plant and microbe interactions. So it includes what's going on above ground and below ground. There are chemical processes, so the soil nutrients that exist in the soil through the mineral components, but also the chemical processes going on between all of the biological components. So plants exuding their root exudates provide a chemical kind of chemical, both nutrients and soil chemistry. They interrelate. And then there's the physical components of the soil. So you have your basic, sand, silt, and clay, which provide the basic structure. But then there is also the organic matter, and that organic matter is constantly being either built up or used up and possibly degraded or destroyed by different land management practices. So when we think about field assessments or lab assessments, generally we kind of think about assessing the soil within these 3 basic categories. So you might have field assessments that really look at the physical component of the soil. You may have lab analyses that look at the chemical and or biological components or the physical components. So we'll kind of take a dive into that in our next slides. So when I started working with farmers who are interested in regenerative ag practices, and also when I started talking to folks who are working on healthy soils grants, a similar question kept popping up. You know, what are some important key either field or lab tests that would be helpful for New Mexico soils, for our dry climates, and that would tell us about the condition or the soil, the health of the soil. And so I started asking that question to try and narrow down all of these options that farmers and landowners might face when they're trying to decide, I know I want to understand my soils and I know I need to assess them either through observation or by doing a lab test, but I don't know what tests to really narrow down because there are so many to choose from. So I started with conversations with New Mexico State University researchers, professors, extension agents, and posed that question. And I reviewed online articles and publications and kind of went through materials that I had on hand. And we came down to lab and field test criteria that included, number one, it needs to be relatively easily understood and interpreted. There's lots of expensive options out there, but they're hard to understand without really diving deep into soil science. I wanted it to be not overly expensive, so something that could be affordable by the public. And then lastly, it could be done with readily available equipment. So you didn't have to go out and buy a kit. It could be with equipment that you might have on hand. So next slide.
Real quick, if I may interrupt, Amy, just if anyone has any questions, feel free to type them in the Q&A or the chat box, and we'll answer them at the end of the session. So onward, thank you.
Yeah, sure. So I came up with kind of a quick and dirty fact sheet that contained lab test options for those interested in regenerative agricultural practices in New Mexico. And on the back, there were field test options also for regenerative ag. And because, you know, we have limited time here, I wanted to select the three that I knew were being done here through Taos Land Trust, and also that show a range of what those tests can tell you. So I chose, you know, your routine chemistry tests, which I think most people are familiar with. I chose to talk about the Haney Test, which maybe some of you hadn't heard of. And then I also chose to look at the biology test, and there are definitely different ways to measure soil microbiology, and one of them is through taking a sample of your soil and extracting the microbes just with water, and then looking directly at what kind of microbes you might see in a slide. So that's called direct microscopy. So you have a special way of counting those microbial communities. And that's different than, say, a PLFA test, which also measures microbial communities, but does it in a different in a different way. But I won't have time to explain the PLFA format. Next slide. And then you might, if you want to get more information on any of these tests, I provided some websites. They include videos, they include online resources, they include different organizations that have information that you can you know, go out and gather more information on specific tests, specific field assessments, etc. Next slide. So first, when we consider whether you're doing field tests or lab tests, you want to first think about what is your soil sampling plan. And it goes back to what is the question that you want answered about your site. So the soil test data that you collect is really only as good as the sampling plan that you come up with. What's really important is to be consistent. So if you do soil testing in the spring and you want to compare your changes in the soil, you want to choose a similar time the following year, the next spring, because there are, especially with microbial analyses, there are changes that are related to seasonal differences. So you might, if you do a soil sampling in the spring and then a soil sampling in the fall, and you see that there are differences in your microbial communities, that may be because what the soil has in the spring versus the fall is different. But if you compare them at the same time or around the same time every year, then that really reflects more the changes in the soil year to year, and it's easier to relate changes in the soil to your land management practices. So if you want to find out more about that, there's more webinars and information that can go deeper into how to do a good sampling plan, soil sampling techniques, and that sort of thing. So next slide. So first, the soil chemistry test. I think most people are familiar with a soil chemistry test. It generally gives you your pH. Here at the Science Center, we have very high pH in 7.8 to 8.3. And then it gives you organic matter. What's your organic matter content? Generally, New Mexico and arid soils are pretty low. So 1 to 2% is typical. And then it gives you, your ratios of nitrogen, phosphorus, potassium, calcium, all of these major and micronutrients that plants need from the soil. And I think what's good to think about is that the limitations of soil chemistry. And I think it's designed to show landowners and farmers what nutrients the soil is deficient in, so that you can then choose to add different amounts of fertilizer. And now we're understanding how the microbiology really plays a role in having plants access the nutrients that are in the soil. And so we're starting to see the soil not so much as what it's deficient in nutrient-wise and then adding that as a fertilizer, but how can we build up the life in the soil to help support the plants get the nutrients that they need. So next slide. So I think this slide captures really well the range of different nutrient cycling and that microbes do for the soil. So the first, on the upper left and the bottom left, you see images of fungi. So on the upper left is a image of fungi basically drilling through rock to extract nutrients from that rock. And on the lower left, you see fungal hyphae weaving in and out of organic matter, really kind of connecting and creating this web that helps keep soil together and helps with aggregation. On the top, the middle photo is a photo of a legume root system, which shows those balls or nodules. And so inside those nodules, bacteria are taking atmospheric nitrogen and making that nitrogen available then to the plant. And then all of that nitrogen also gets released to the soil when those roots die and picked up by other legumes. The upper right image, thank you, shows, that's a picture of plant root hairs, and it's showing that there are bacteria that are going inside the root itself, the tip of the root, And there is some exchange between the plant and those bacteria, such that when the bacteria are released again by that plant root, they notice that there's a change in the membrane of the bacteria. And so they're thinking that maybe that there's some nutrient exchange happening between the bacteria and the plant root. Again, you know, there's all kinds of ways that plants are able to get, you know, access nutrients through all of these intricate relationships with microbes. The middle image on the bottom is a picture of an acari, and those little microarthropods go around and shred up organic matter and make them available to other smaller microbes that, you know, take those nutrients and you know, continue decomposing. And then on the bottom right are two pictures of amoebae and compost. So they're both testate amoebae. One is dividing. It's in the process of making a new shell, and then once the shell is done, then the amoeba will populate that shell, and there will be two. So there's just so much else beyond what I think our typical chemistry tests can show us in terms of how plants can really thrive in the soils. So next slide. And then the last test that I know that the Taos Land Trust is also working on is assessing the soil through direct microscopy. So taking a small amount of either a field soil or a compost or even a compost tea or extract and looking at it under the microscope and doing counts of what kinds of microbes you see. So you might see nematodes, which is the image, the big image, large image to the left, or you might see fungal hyphae to the right, or you might see these little rotifers that go and they're grazing the bacteria and really helping cycle those nutrients. And then on the upper left is a fungal strand that was an image from the compost that Taos Land Trust is producing. So all kinds of, you know, microbes that are out there doing lots of great things for the soil. Next slide. Oh, and then also there's the Haney Test. And so that's a combination of biological data and chemical data. And really the Haney Test is designed, as it says, to mimic nature's approach to soil nutrient availability by looking at both biological and chemical test data. And the way they do it is they will quantify different food resource pools, either through a water extract. So they'll look at nitrogen, carbon, and also your C to N ratio. And then they also compare that with a weak acid, which is supposed to mimic what root exudates will be able to extract from the soil. And so next slide. So when they have these different food resource pools, they also compare that to what is the microbial activity that's happening in your soil. Or that's to say, do you have enough microbes there to really process and cycle all of those nutrients and make them available to the plant? So in summary, the Haney Test measures the quantity and quality of your food resource pools, and it relates that amount of food that's available in your soil to the microbial ability to cycle those nutrients and make those plant microbe associations. And lastly, they look at what is the C to N ratio of the soils. And that really helps determine whether your soils are balanced between carbon and nitrogen, and whether or not your nitrogen might be tied up by excess carbon, for example. Next slide. And then the last thing I can say is that it's really important, if you're interested in understanding your soils, is to go out and look at your soils and look at them in a measured, kind of regular, consistent way. One of the things that we started doing here is comparing our different fields to what kind of earthworm counts we're seeing in different fields. You can measure infiltration, which is the soil's ability to absorb water. Are there areas of compaction? Is after a huge rain event, are you getting ponding? And then also make sure you dig a hole and look at the soil below the surface and see, how are the plant roots growing? Are they, you know, is there topsoil? Do you have any topsoil, or is it not measurable? And then lastly, what is the surface of your fields like? Is it covered, as Ben was talking about? Do you have enough adequate, you know, living cover or mulch to protect your soil from those heavy rain events and protect it from erosion? Those are all things that year after year can tell you if you're moving in the right direction or not with respect to soil health. Next slide.
Okay, thanks, Amy. And just want to show briefly exactly what we did here at Rio Fernando Park. As Amy described, we sent off soil samples to do the basic tests, so the chemical test. And you know, that gave us some understanding what was going on, but it was you know, it's a little abstract just to read the numbers of the different chemical constituents of the soil and actually what they mean. It takes some study to figure that out. More on hand, we did some physical tests, and these are UNM CCC interns doing some of the testing. On the left, we have a device called a penetrometer, and that measures the compaction in the soil. Basically, you push down on this rod into the soil, and the harder that you have to push, the harder it is to get it into the soil, but then that dial cranks up to the right into the, or clockwise, I should say, into the red, and that you know that it's more and more compact. And compaction is really a major problem for soils, or at least if you want to grow plants on them. I mean, if you want to build a house on your soils, then you want as much, you know, you want a lot of compaction. But if you're trying to grow plants, you have to realize that soil is made up not just of the substance of particles, but also the spaces in between. And so those spaces are occupied by air cavities and water cavities. And assuming it's rained recently or irrigated. And those cavities are where the plant roots go to find not just water and air, but also to interact with the microbes, and so that they can participate in the nutrient cycling. So compaction really prevents the ability of plants to take up nutrients, and it really prevents the ability of plants to grow good root systems, and obviously they will suffer on top. And to the right, that's a device called an infiltrometer, and that measures how quickly the water soaks into the soil. And that's related to compaction, but a little bit differently, because it kind of depends on the soil structure, how quickly it goes in. Like a sandy soil, the water will disappear immediately. If you have more clay in your soil, it'll take some time. But also if you have compact soil, it's going to take a long time. So we took measurements of both the infiltration and the compaction and recorded those, and hopefully our efforts will see those numbers improve. The last physical test we did was aggregate stability, and this is just a simple test where you take a PED or a cloud of soil and you immerse it in water, and then you time the rate that PED takes to dissolve into solution. In some cases, you'll see that, you know, it'll barely be disturbed by soaking in the water at all. In other cases, it'll dissipate almost instantly. And that tells you a lot about the types of organisms that are living in the soil and the exudates, as Amy described. And then lastly, we did some various forms of biological testing. We sent samples, both of our field samples and compost, off to labs so that they could look at it through their microscopes, since we're just kind of beginning in that department. And then we also did the Haney test, as Amy described in some detail, and the PLFA test, which is, she didn't really talk about, but that's the phospholipid fatty acid test. And that basically measures the microbial activity, respiration. And so fungal and bacterial primarily, but other microbes living in the soil. And then we made attempts at our own microscopy and, just began the process of being able to identify what's living in the soil. We're hoping to gain those skills into the future. And then, so, we're gathering all this information. So then what do we do about it? And so really the goal is not just to know, but to actually improve the soil health. And, I've been to a lot of workshops and done a lot of reading about just the importance of grazing animals for healthy soil projects. But for various reasons, we're choosing not to do that at this time. And but we're also becoming creative in other techniques that will yield the same results. So over the last 3 or 4 years, we've engaged in P-line plowing, and we're not doing any tilling of the soil. We're not turning it over at all. We're not disking. We're simply working from the top down with mulches and living crops. All the seeding that we're doing is through a no-till process. So it's with a tool that basically inserts the seed right below the soil surface and covers it up. And so again, the idea is to not disturb the microbiome that is living just underneath. We're working with diverse species of cover crops. So not only legumes, but also grain crops, which are quite good at suppressing the weedier crops. And they also are very strong exudate releasers, which feeds the microbial communities. And then, even wildflowers and native grasses to work with the pollinators that are present on the land. We're, as much as possible, we're trying to keep a living root in the ground through living mulches. And if, in the winter time, we just want to make sure that we have something covering the bare ground. And then we're very active in making our own compost, mostly out of materials that come from on site. And over the past 3 years, we're getting better and better. I wouldn't say that, we have it down to a science yet, but we're learning as much as we can and engaging in new techniques and just ways not only to make the compost, but to test it and then also to apply it on the land. And Amy's going to give us a little more detail on the one method of making compost.
Yeah, so this Johnson Sioux bioreactor compost, I'll give you a little preview. I think the compost process is different, and I think the result ends up creating a compost that's really rich in microbial diversity and also high in numbers. So how does that happen? It basically revolves on just a few very simple principles, and I will preface this by saying, all of these materials you can get at the hardware store for very, relatively inexpensive, and these can be built in a day or two once you have all the supplies. So it's pretty, once you have all the materials, it's pretty simple to kind of do it yourself. But the compost process itself relies on a few simple principles. One is you really don't want to disturb it. Once you fill it once, and you leave it undisturbed for 12 months, which is a pretty long time for most compost. I've worked in commercial compost before, and we're trying to finish compost between, 30, 60, 90 days tops. So we're really trying to get the composting, the decomposition process going quickly. The second principle, besides don't disturb it, is make sure that it's aerated, and I'll explain how that happens. And then the third is make sure you water it. And Dr. Johnson here on the left, he's a researcher at NMSU. He and his partner, wife, Wei-Chin Su, developed this process together. So leave it undisturbed, water it daily, make sure it's aerated, and if at all possible, try and make sure that the compost doesn't freeze. So basically, the way you build it is you get a wooden pallet, and you cut holes that will fit a 4-inch PVC pipe down to the bottom, and these will serve as the aeration channels for the compost. And once you have those cut in the pallet, then you lay woven landscape fabric over the pallet with those holes cut out so that the compost doesn't drop through the slats. And then the wire cage is basically a concrete remesh. It's 6-inch grid, I believe. And you kind of roll that and tie it together and fit it on top of that pallet. And in the inside, you line it with, again, the woven landscape fabric. And that's a breathable material, but it will hold the compost inside so it doesn't fall out. And then lastly, you have some 4-inch PVC perforated pipes. So they have those holes. They're actually the sewer drain pipe PVC. And you stick those in the holes that are indicated on the pallet. So once you have that constructed, you're ready to fill it with your compost materials. Next slide. So on the left is an example of the materials that I used for one of my first composts. So there's wood chips, there's cottonwood leaves, there's alfalfa hay, there's food waste. I added a little bit of composted dairy manure just to see with some added organic nutrients. And I think that's about it. And then the next step is to make sure that your materials are well saturated. So we have on the bottom left, Rob Hyduk, who's dunking some 5-gallon buckets that have holes for the water to drain out, but making sure that that material gets soaked all the way through and it's wet when you load it into the bioreactor. And then on the upper right, we have an example of, going ahead and filling that with trying to keep those PVC pipes, vertical. And then at the bottom, we have a finished filled compost bioreactor. Now those pipes only stay in there for maybe 1 or 2 days, and then you can lift them out and those aeration channels will stay intact. Next slide. So, and then you're gonna let it, once you filled it, you're gonna let it sit. And so in the first few days to a week, the temperature in your compost is going to rise, and it will rise depending on what the recipe you added it, or the recipe of materials that you added to your compost. So compost typically has, a heating phase where it gets really hot. It might cool down a bit, heat up a little bit more, but eventually it's going to start, the heat is going to start to dissipate and it will cool down eventually to ambient temperature. At that point, when it cools down, if you'd like to add earthworms, you can certainly do so. They help keep the compost churning, they help keep it aerated, and of course they add wonderful worm castings to your compost. And so you can see on the upper right, the compost that I'm holding, I think is almost mature, if not it's maybe at 11 or 12 months, it still has, you know, quite a bit of wood chips. So the wood chips that I added didn't completely decompose. So what I did was I sifted those wood chips out, and we used the wood chips for any kind of wood mulch. And then what you see on the bottom right is the sifted compost that's after 12, at the 12-month mark. Next slide. I did have an irrigation system. It's if you follow the line on the bottom, you hook up a hose to the bottom left, and it actually, you know, ran through all 4 of my bioreactors, and then it runs down to little emitters. So the water kind of is a mist, and I emitted it, or I watered it for about a minute every day, which it ended up being about a gallon a day. And then, you know, I didn't have my compost protected in the winter, and it did end up freezing, so we didn't water it when it was in those months when it was too cold. So again, you know, just make sure you water it. Make sure the aeration is maintained. Don't disturb it. Don't turn it. Try to avoid freezing, and yes, add worms, because it will definitely improve the quality of your compost. Next slide. So one of the benefits of having the process or the compost, you know, have a moist environment, be undisturbed, is that this environment favors fungal communities over bacterial communities. And so I'm just showing a couple of pictures of some fungi that grew in my compost. These appeared after the heating phase ended, and it started to cool down. But you'll notice, you know, you may have compost, you may have evidence of fungi, and that's a good sign that your compost is on the right path. Next slide.
Amy?
Yes.
Sorry, it's Susan. We have a couple of questions. May I ask the 2 for you?
Sure.
One is, how do you keep it from freezing in the winter time?
Right. So David Johnson recommends that if you have a greenhouse, you know, or a hoop house, you can keep it in there if there's access to water. Another suggestion he recommended was, you know, you can line it with hay bales and kind of keep it protected then. The bioreactors, the compost that I've done here at the station, I didn't have, we do have hoop houses, but it wasn't good access to water, and they kind of would be in the way of other research experiments. So mine did freeze, and I did, you know, look at it under the microscope the following year after the 12-month period. And I didn't see, you know, as good of microbial diversity as, say, Dr. Johnson does, but I was definitely seeing nematodes and fungi and bacteria. I was seeing a good diversity of organisms in that compost. So I think avoiding having it freeze gives the microbes that extra, you know, 3 months to really develop, to continue developing. When the compost freezes, they're still there, but they're not able to kind of continue developing those numbers and microbial diversity. And David Johnson says that yes, it does set back your diversity a bit when the compost freezes.
Okay, here's another question, if you can do this one too. Can you explain the difference between compost, especially Johnson Sioux compost, and chemical fertilizer?
I would describe the Johnson-Su compost as the whole idea is that, you know, when we think about compost, we think about a nutrient amendment for the soil. And I think the difference with the Johnson-Su bioreactor compost is, yes, there are nutrients. It is a high quality compost, but I consider it more of a microbial inoculant. So the whole idea of giving it time, giving the bacteria, the fungi, the nematodes, this time in an environment that's undisturbed, you really create a rich ecosystem that's allowed to develop. Whereas in other composting processes, it's turned so often, it's managed so often that really those delicate organisms like fungi aren't allowed the opportunity to get a foothold. And so what you have is more bacteria or more just organic matter. So I think Johnson-Su bioreactor compost really, you're able to have a product that has life, you know, all of the different organisms that may not be present even in our soils, they can be grown and developed in our compost.
Okay.
So it kind of ties into this last slide, where the idea is that if you can develop these microbial communities in your compost and then either use the compost or use the extract, as Ben is going to describe in a minute, and re-inoculate your soil, then it's a chance to kind of help kickstart your soil and help your plants reestablish those relationships with the microbes. And this picture, I think, shows well that really the microbes are interdependent with the plants. You know, they really, there's a symbiotic relationship established between all of these beneficial organisms and the plant. But that also, as you know, we were describing earlier, that above ground processes, animals grazing, the insects, the birds, all of those organisms have an effect also on below ground. So it's very complex. So next slide. And so I did want to show, I know these slides are impossible to read, so it's just really a visual. So Ben or Taos Land Trust did send a sample of the compost, the bioreactor compost that they were developing, and they also sent in a field sample, and that's from their grain trials. And I just want to note that just to compare, when we're looking at fungi, the flagellates, which are protozoa, amoeba, which are protozoa, or nematodes, you see the compost really have you know, higher levels. All the you know, the data that's shown in green are good levels. The data shown in red in the fields are deficient. So if we can inoculate our field, say with the fungi and the flagellates and the amoeba and the nematodes that come from our compost, and if they get established well in our fields, then the plants will kind of have the opportunity to kind of continue growing these microbial communities on their own. So that's what we hope to see. Next slide.
Okay, so we have put 9 of these Johnson-Su bioreactors into motion. The image on the left is that's 9 of them in place. We intend on building a hoop house over it, which probably won't keep it from freezing in our climate completely, but you know, might give it just a little more length to the season of it. But what we did, this is what these reactors here are filled currently for next year's round of compost. Last year we did 4 of these, and we took that compost and we turned it into an extract, which was then sprayed out onto the fields. So the process of that was that this compost was taken and put in the bucket here with water, and it was mixed into a slurry. And then that slurry was then added to this tank here, and it was thinned down even further, such that it was able to be sprayed with the sprayer from the tractor. So we sprayed it on 2 acres so far, and we're planning on doing 4 more acres this year. But one thing that we did as we went through all these stages of compost, we took a look at it through the microscope, and we wanted to make sure that we weren't destroying the microbes, which were so important in the compost, by spraying them out on the field. And these tests are still rudimentary, and we have a lot to learn, but this was our first stab at it. And I mean, one thing that going through this Johnson-Su bioreactor process, from here all the way out into the fields, is I realized that the compost, it's not the material of the compost that's important. The compost is actually a substrate for all these organisms to live in. And what's really important is that we get the organisms out onto the fields where they can then form associations with the plant roots. So it kind of altered my view of, really what we were feeding. I mean, we're like somebody asked earlier, the difference between fertilizer and compost. And I think of fertilizer as something you normally would use to feed the plants, whereas compost, you're feeding the microbial community, which then forms associations with the plants. So it's kind of more of a holistic way of improving the health of your plants.
Ben, can I ask 2 questions we've gotten?
Absolutely.
A viewer writes, I am assuming Alcalde may not have as long of a freeze season. North of Taos can be pretty long. Can you just let it sit unwatered for a few weeks? I don't have a way to keep it warm.
Well, there is a difference between Alcalde and Taos. It's not as much of a difference as the difference between Taos and Las Cruces, where Dr. Johnson is doing a lot of his research. So, in talking with them, he admits that we are a little bit more challenged in, protecting our compost from freezing. And, so definitely everything we can do to keep them from freezing is better. Certainly in the winter when the compost is frozen, there's no point in watering it because water, just turns into ice. So, I mean, we haven't watered ours through the winter. It's frozen, solid as a block of ice, and then defrosted in the spring, and then continued its process. And as Amy described, the microbial communities do take a hit, but, they are still largely viable.
Excellent. And then one more question. What are living mulches and surface residue that you mentioned earlier?
So living mulches are basically cover crops, they're also described as, and they're plants that are planted for their benefit to the soil. And there's, it's a cocktail of different plants. And so the legumes are fixing nitrogen in the soil or attracting the types of microbes that fix the nitrogen, I should say. You have the grains, which by their physical height, they prevent light from hitting the bare ground, which might inhibit the germination of weedy species. The grains are also known to exude the sugars that the microbial communities feed on. And then, there's a variety of other functions. Sometimes people put daikon radishes in their cover crop mixes because the radishes grow so long and deep that they break up the hardpan in the soil. So, that's kind of a physical characteristic that we're working on. So living mulches are basically cover crops that, have an intended function for the soil health. Was there another part of that question?
Surface residue?
Oh, surface residue is basically, well, it's difficult to keep a lot of plants growing through the winter for the reasons mentioned above. But, a lot of times we'll cut the plants down in the fall, and the mulch that lays on top is the surface residue. So it's just basically, like, for example, we had rye grain growing here, and we harvested the rye seeds, but it left quite a bit of straw, which we then mowed down and laid on the surface of the soil as a kind of a soil armor, sometimes people call it. And so, you know, the residue is just the material that's left over basically after the plants themselves have died.
One more question came in, Ben and Amy, and that is, would you recommend starting a bioreactor in the fall so it has a good chance to survive and thrive through the next summer months?
I'll let Amy go for that one.
I think the best, I think you can do it in the fall. I think it's better to start your bioreactor in the spring or the summer, and that's just because it gives the bioreactor a longer time to really develop those microbial communities before the winter hits. If you have a way to keep it from not freezing through the winter, then starting it in the fall is fine. And I have started some in the fall. I just think they do a little bit better if you give them more time to develop before the winter.
Thanks, you guys.
Mm-hmm.
Thank you. So thanks for the questions, by the way. I'm glad to know that you're out there, because it's a little strange talking to all of you and not actually seeing your faces. Just to continue on, so we've been doing all this work. I mean, not just over the past year, but you know, for the past 3 years, and you know, we're making some improvements. But this year we have some more work to do, and I was just discussing that with Amy. I mean, we've done a lot, but there's quite a bit more to do. We have seeded already the pollinator habitat. We did the compost extract, as I described, and the seeding. So that's ready to go for next year. But we still have 4 acres of grain areas to do the compost extract and then seed with, we're doing 2 acres of rye and 2 acres of winter wheat. And you know, but the seed is going to be mixed with the cover crop. So we have a whole cocktail going out there. And then ideally, if we get the water we need this winter, then we'll be harvesting wheat and rye next year about this time. And then we also have another round of soil testing to do, because the grant actually requires 2 soil tests at the beginning of the season, the end of the season, to see if some of the measures that you're enacting are showing improvements. So we'll see about that. And we have to do, in the last year, we realized that, well, I mean, we've known it all along, but I guess the importance of it has come to a head, is just that water conservation is really key to doing what we want to do with the land here, because this past summer we barely had any water out on the land, and so we didn't really have much of a crop to harvest. So we really have to you know, just look to examples of people growing food in a lot drier places than this, and you know, just understand the ways of taking care of the soil such that it becomes much better at infiltration and much better at making good use of the water that's available. I mean, there's not going to be more water than, you know, that the weather can produce, and we can't certainly push taking more water out of the ground either, because we're really interested in working with what we have. So I just want to let you all know that this is part 1 of this webinar, and part 2 will be coming up in a few weeks, and I will be sure to let you all know. In part 2 of the webinar, we will be discussing more about the no-till seeding and more information about the cocktail of cover crops that we're working with. And we're also going to discuss more about water conservation and some of our plans for next year for how we're moving forward. So with that, I want to thank you all, first of all, for attending. And thank you very much, Amy, for your really good talk and the detail and all that. And everyone else listed here, I want to particularly thank Susan Ness for being our manager and our chat monitor. If you have any further questions, feel free to email me. And if there's questions for Amy, I can pass that on to her as well. But
there's a question for you.
Okay, absolutely.
Yeah.
Go ahead.
Well, how do you harvest the grains if they're mixed with cover crops?
That is a good question, and the answer is that typically the grains. So I specifically chose cover crops, and we'll cover this more in the next session. But I chose cover crops that were lower growing. For example, I left out the triticale in the cover crop mix because the grains will grow taller in general than the cover crops. So we should be able to harvest above the cover crop. But you know, it's still an experiment, and you know, we're hoping that all works out. But yeah, so the cover crops are generally going to be lower growing. The grains are growing taller. So that's the short answer to that question. So with that, thank you all for attending. And oh wait, there's
Let's see. Oh no.
I see more. Okay, we got it. Okay, thank you all. I'm gonna sign off now.
Thank you.